Today we present a paper from Cell Metabolism titled Gut Microbial Metabolites Facilitate Anticancer Therapy Efficacy by Modulating Cytotoxic CD8 plus T-cell immunity. Gut microbiota have been demonstrated to play a crucial role in tumor immunotherapy. The gut microbiome synthesizes numerous specific metabolites, and recent findings indicate that certain microbial metabolites can influence cancer progression and modulate systemic immune responses. This study demonstrates that the gut microbial metabolite, butyrate, enhances antitumor CD8 plus T-cell responses via IT-dependent Ilmenus-12 signaling. Combination therapy with butyrate in either chemotherapy or immunotherapy exhibits enhanced tumor suppression, suggesting that supplementing gut microbial metabolites may have implications for future cancer treatments. First, the research team treated mice subcutaneously implanted with MC38 colon cancer cells using three approaches: an antibiotic mixture ABX comprising ampicillin, neomycin, vancomycin, and metronidazole, oxaliplatin, and ABX supplemented with gut microbial metabolites. Results showed that oxaliplatin controlled MC38 tumor growth, but ABX treatment significantly suppressed its efficacy. Interestingly, mice treated with ABX supplemented with gut microbial metabolites exhibited marked tumor regression after chemotherapy, suggesting the gut microbiome may influence cancer treatment responses through its metabolites. Focused examination of CD8 plus T cells revealed that oxaliplatin treatment induced a dramatic accumulation of CD8 plus T cells in the tumor microenvironment TME and enhanced IFN production by CD8 plus T cells from draining lymph nodes DLNS. Oral ABX treatment nearly completely suppressed this response. Oral administration of gut microbial metabolites to ABX-treated mice was sufficient to rescue CD8 plus T cell tumor infiltration and promote IFN production fade 1C, 1D, S1E, S1F, suggesting gut microbial metabolites may enhance chemotherapy efficacy by promoting CD8 plus T cell responses. Next, we tested whether CD8 plus or CD4 plus T cells were essential for the tumor regression induced by microbial metabolites. Interestingly, depletion of CD8 plus T cells, but not CD4 plus T cells, eliminated the full anti-tumor effect of gut microbial metabolites, indicating that CD8 plus T cells play a critical role in the tumor suppression mediated by The research team compiled a catalog of gut microbial metabolites significantly reduced in ABX-treated mice and screened their effects on CD8 plus T cells. Among 63 metabolites, butyrate most potently promoted IFN production by CD8 plus T cells, suggesting butyrate may mediate the anticancer effects of gut microbiota. Preventive oral administration of butyrate in drinking water also improved cancer progression and rescued the CD8 plus T cell response. Interestingly, butyrate monotherapy failed to control tumor growth, indicating that the anti-tumor effect of oral butyrate depends on chemotherapy. Collectively, these data demonstrate that butyrate modulates anti-tumor CD8 plus T cell responses and may be beneficial during chemotherapy. The research team first examined the survival and proliferation of butyrate-treated CD8 plus T cells. Results demonstrated that short-term butyrate treatment significantly enhanced the cytotoxic capacity of antigen-specific CD8 plus T cells, while increasing granzyme B production. To determine whether butyrate could also enhance CD8 plus T cells in vivo, OTI-CD8 plus T cells stimulated with butyrate in vitro were transferred into EG7 tumor-bearing mice. Ex vivo butyrate treatment significantly enhanced the in vivo anti-tumor activity of OTI-CD8 plus T cells, markedly increasing both intratumoral accumulation of CD8 plus T cells and IFN production. Collectively, these findings demonstrate that butyrate treatment directly modulates CD8 plus T cell function to promote anti-tumor immunity. RNA sequencing results indicate that a pair of transcription regulators, A2 and E2A, are significantly regulated by butyrate and CD8 plus T cells. It can block E to A binding to DNA and antagonize E to A transcriptional activity, while ID2 and E to A participate in the differentiation of numerous immune cells. Their roles in tumor-infiltrating lymphocytes TILs remain unclear. The research team examined ID2 expression levels in CD8 plus T cells under various conditions. Interestingly, ID2 expression was markedly higher in tumor-infiltrating CD8 plus T cells compared to naive, activated, or memory CD8 plus T cells. Subsequently, MC38 cells were inoculated into WT and ID2 mice, followed by treatment with oxaliplatin. Data from mice subjected to both treatments demonstrated that the anti-tumor efficacy of butyrate depends on ID2 expression in CD8 plus T cells. SCRIS-seq data indicate that ID2 deficiency in CD8 plus T cells also leads to significantly reduced IL-12 receptor expression. When WT and ID2 CD8 plus T cells were isolated from immature mice and treated in vitro with CD3 divided by CD28 alone, they showed no significant difference in IFN and granzyme B production. Although butyrate monotherapy significantly enhanced IFN and granzyme B production in both WT and ID2 cells, butyrate induced greater IL-12 expression in WT-CD8 plus T cells compared to ID2 cells, suggesting butyrate modulates IL-12 expression via ID2. IL-12 treatment, particularly IL-12 plus butyrate, significantly enhanced the WT-CD8 plus T cell response, but failed to do so in the ID2 group. ID2 promotes cytotoxic function of CD8 plus T cells by regulating IL-12 signaling, and butyrate enhances CD8 plus T cell function, modulating the ID2-dependent IL-12 pathway. Furthermore, ID2 is essential for IL-12-induced CD8 plus T cell proliferation. IL-12 blockade significantly suppress butyrate-treated OTI-CD8 plus T cell tumor infiltration and anti-tumor efficacy in vivo, indicating that butyrate-mediated anti-tumor effects depend on IL-12 signaling. Since ID2 can bind to E to A and antagonize its transcriptional activity, we next examine whether E to A regulates IL-12 receptor expression. Chromatin immunoprecipitation and luciferase assays revealed that E to A directly binds to the IL-1R, the II locus, and suppresses its expression, with ID2 deficiency further enhancing E to A's inhibitory effect. Collectively, these data demonstrate that butyrate in ID2 promotes CD8 plus T cell anti-tumor immunity by modulating IL-12 signaling. In summary, this study demonstrates that the gut microbial metabolite butyrate can modulate anti-tumor CD8 plus T cell responses through ID2-dependent IL-12 signaling. This indicates that gut microbial metabolites can enhance anticancer immunity, thereby significantly improving therapeutic outcomes. Consequently, gut microbial metabolites may serve as an effective component of cancer treatment. That's all. Thank you for your attention.